A scientist, Rosalind Franklin, used the X-ray diffraction to define the double helix structure of DNA. He discovered that there are two strands of phosphate sugar backbone with nitrogenous base twisted together. Every single unit of DNA is nucleotide, which consists of one deoxyribose, a kind of sugar, bonded with a phosphate group and a nitrogenous base opposite to the phosphate group. The model of DNA was first constructed by Watson and Crick in 1953. Okay, let's look at DNA replication process. When eukaryotic cell doing replication, the process of DNA replication occurs in S phase of the interphase, and this process occurs in nucleus. In 1958, there are two scientists named Michelson and Stoll conducted an experiment with N14 and A15, which is highly reactive isotope of nitrogen, to identify the replication pattern of DNA. Well, they first put E. coli into N15 medium for well, then transferred the bacteria into N14 medium, and centrifuge. They find what appears in the centrifuged second generation E. coli is two strands of DNA, which one of them is purely consists the N14 and the other is a mixture, which proves the semi-conservative replication of DNA. Now, the DNA replication. The first step is to unwind the double helix structure of DNA. Helicase is a kind of enzyme that uses ATP to move and unzip the two strands of DNA. Single-strand binding protein attached to each strand keeps them open. DNA polymerase III catalyzes the formation of new strands using DNTPase. DNTPase stands for deoxyribonucleotide triphosphate. By pairing the DNTPase to their complementary base pair from 5' to 3', the leading strand was formed. On the opposite strand, things become more complex. DNA structure is antiparallel, so if one strand of DNA molecule is from 3' to 5', the opposite one will be 5' to 3'. But the enzyme DNA polymerase III only reads from one direction, which is from 3' to 5'. So a discontinuous transcript occurs. The segments created are called the Okazaki fragment, and the strand is named the ligand strand. At first, the DNA polymerase III will replicate a short episode, then it leaves. RNA primer will put a short episode of RNA onto the template DNA as the starting of transcription. Then the DNA polymerase I replaces the RNA primer with DNA segment by transforming RNA long range into DNA long range. There will have a little gap between them, but an enzyme can cope with it, which called ligase. It fills the gaps between Okazaki fragments, and the new strand was formed. Well, we could actually artificially synthesize DNA in labs. PCR is a technology used to elongate the template DNA strands and made a million copies of it. The most valuable application of PCR in medical laboratory is the diagnosis of infectious disease. Chromosome is composed by protein and nucleic acid. How can we define the DNA as the hereditary material, but not the protein? Hershey and Chase used bacteria and virus to determine it. Because DNA contains phosphorus but not sulfur, which protein contained. Through this way, we can verify DNA is the hereditary material. We call it Hershey-Chase experiment.